dive plan here, just to look at this. All right, so where are we?
Tilt up on your camera just a bit there.
Tilting up.
Yeah, that's good, thanks.
Okay.
This is steeper than I imagined here. I thought it was going to be a nice gentle 20 degrees the whole way, but...
There was we kind of spotted this feature on the sonar. See this little kind of wall here?
Yeah.
That was first feature I've seen. I don't think it's very big, but it's a wall to look at.
Sometimes on these vertical surfaces you can find high density communities, but
yeah.
Sometimes if the substrate isn't all that stable, or if the currents aren't right, you won't find that density. But sometimes they can be absolutely carpeted with biology.
Might have time to follow it back to the right here. I'll come up to the top of it. Maybe we'll... Come up just a bit there.
Coming up.
That's the view we want right there. Beautiful.
So would these classify as pillow basalts still?
Yeah. Just smaller, like 1 meter sized pillows. And some of them don't look like pillows, but that's just because they overlap over and over and over in layers. Sometimes the pillowy morphology, I suppose, would get lost, but
That's a little pillow outcrop there.
So our last primary rock sample was 3156 meters. We've moved up to quite a bit. I don't think there's a particular interval we're necessarily interested in, but as we, the next 100 meters or so vertical, maybe we can take another rock, 2,500 or so.
Copy that. Wasn't that every 500-ish or something like that?
Yeah, something like that.
You can zoom in there a bit if you want, Dan. Can't quite tell what that guy is.
Glass sponge. This one's in the family Pherididae, almost certainly. Some associates there, some kind of shrimp maybe. Can just make out the red of the carapace.
I touched the rock there. Okay, can go wide.
Yeah, looks like there's some fairly large sponge rubble too.
Sponge rubble.
Yeah, sponge rubble. So oftentimes the stalk of sponges, of these big, long glass sponges will fall down and get stuck in the rocks underneath, and sometimes it can persist for long periods of time. This is usually a good
Yeah.
indicator too that there's stuff upslope, living stuff. So whenever you see debris and falling material, that usually indicates that there's probably a good concentration of things that's upslope and persistent.
Fascinating.
Or it could just be a really good collection spot. But since these don't really look, it doesn't look too much, valley-like. It's these ledges are capturing any fallen debris material. Suggests there's probably more upslope that we haven't seen yet. This one looks like another stalked sponge, but the sponge seems to be gone or missing or dead, and it's been recolonized by a couple of crinoids here, using the stalk as a way to get up into the water flow so they can feed more easily. They're suspension feeders as well.
So would those crinoids basically attach themselves to the dead sponge, or?
Yeah, they have structures called cirri, which are kind of like feet or legs that it uses to attach and wrap around branches of things, and they can hold on pretty well. They can release them and swim off, but for the most part, once they're attached to something, they don't move very often. And the stalked ones are completely incapable of moving, although there's some evidence that some families of stalked crinoids can move little bits, or they can detach, and that's another way they can move if they need to. But most of the time, you don't find them detaching without a good reason.
You want to go up the hill, the local hill
Yeah,
here, Steve?
yeah, let's do it. Yeah, there's.
Let's change her bearing to. Let's try east 090. Split. Sure.
There's nothing particularly sacred about the direct line between the waypoints. We can investigate whatever we'd like. It seems like there was a fair bit of density of sponge rubble in this direction that we're headed now, so hopefully we'll see some more high density sponges as we move upslope. It's touching the buttons on the bottom. That's why this thing is popping up.
I see.
Can you take a quick zoom on this branch when you have a minute?
Yeah. Go ahead, Dan.
This was formerly a coral, but it looks like it's a dead bamboo coral colony. You can see very easily without the tissue here the node points there. This one's a little calcified over node point, but no living tissue. So this one might have been either eaten or somehow died off. But what's surprising is that hasn't been really well colonized. There's a couple of hydroids and things, small colonies of cnidarians, but nothing too large like crinoids or barnacles at this depth. Thanks, all set there.
How about 135 for 50 meters? see what happens. Yeah, yeah. Just tell her 135, 50135.
So if you're looking on channel 1, the 2 green laser dots, those are 10 centimeters apart. Can we look down here first? 10 centimeters apart, those scaling lasers are useful for telling the size of things in the seafloor,
Push in there then.
where our perspective is a little bit distorted.
Zoom right in there if you want.
So we have here, it's a
Gibson, you can
golden coral in the genus Chrysogorgia.
come down 5 meters.
It has,
Down 5.
it looks like a pair of squat
Change your heading
lobsters.
to 135.
At least one, maybe.
I tried, but it's
One squat lobster.
Squat lobster.
Yeah, right in the middle.
Come down a little more.
And those squat lobsters are often live with this this colony its whole life. This one is fairly easy to identify. This might be Chrysogorgia chryseus, which is kind of this, has this what's called
Okay, it can go wide.
biflobellate colony, so it has 2 branches, and it kind of forms like two halves of the
if I
colony,
change to
and then the
135
squat
here.
lobster would live in the middle.
the thruster.
It has these very unique
come around.
star-shaped polyps. They often look like
I'm
very
pulling on you
spiny
for a moment.
stars. They're in their own clade within the Chrysogorgia species, different than the ones we saw a bit deeper, the bottle brush types, different clade. But pretty common at the species that's fairly commonly observed at this depth,
Wow.
although not as abundant as other places we've seen it.
You said this was a golden coral, correct?
Right.
So would they calcify their skeleton almost similarly to black corals, or quite
We're good on the delta there.
It's similar skeleton. So the difference
I'll
is
stop
that
on the wedge,
the
Gibson.
golden corals have a calcium carbonate skeleton that's made out of a mineral called calcite, or high magnesium calcite, and the black corals have a protein-based skeleton.
Ah, I see.
It doesn't have any calcium carbonate in it.
What's happening? You got plenty underneath you, but
Yep.
I'm
Way it's stretched out.
yeah. So in that case, I'll be the delta will be in some
Small.
cases negative.
Understood.
But yeah, so we're keeping the same kind of distance.
Yep.
But since I'm way out, you got to come down lower. And
This is Chrysogorgia, possibly chryseus. There's also a very similar looking one. But
Argus
this one looks most similar to chryseus. There's also a fairly similar looking one called stellata as well, but it tends to be bushier.
Do you want me to pan up to get the view there?
if you want, yeah, you can pan up a little bit. It's actually pretty
Both
good
found
there.
at similar depths, though. Sometimes it's very hard to tell them apart. They both belong to the same clade, but genetics, for example, would be able to allow us to tell them apart fairly easily.
Fascinating.
What's weird? Yeah, it's because she's changed her bearing and dressed in sideways there, and then Avalanche is quite down the slope from.
Is that a 20-meter spacing on the grid, Lynette? Yeah, it's really jumping around. Oh.
We're noticing more density of these unbranched, very long bamboo corals. Right at this depth, as we get a bit shallower, another Brusingid in the lower left. But the bamboo corals are definitely dominant here.
Be a good time to pick up a rock, Steve. We're kind of waiting for Atlanta to change direction there.
Yeah, does anything look good in this area for angular good rocks?
Yeah. We
Yeah,
just did it.
I think where the lasers are right now look okay.
In that, like, kind
Yeah.
of pile?
Yep.
What do we have open for boxes? Starboard boxes. Probably got a few spaces left.
That's the way we're going, yeah.
Like port
Happy with the...
on the
Yeah.
forward bio box.
She's still
I think
got
we'll try
10
and
meters left
so
on
that
the move.
it's easier to put some like of the fine squishy stuff like the biology in the forward box. We'll try and load up the starboard box first as much as possible.
Okay. Yeah, we have 2 spots left.
We can also these outer bins are sometimes large enough that you can double or triple up, but at least double up for sure. I don't think we're taking anything boulder size that would take up the entirety of the box. Oh yeah, feel free to use the circle what you think is appropriate.
This guy?
Oh, I was looking at
Okay, yeah, what are you looking at?
that guy, but you were looking at what guy? This guy? That guy?
Yeah, that guy.
This one?
Uh-huh.
Roger.
It looks like there's some organism on there.
Oh yeah.
Oh, nice one.
Too.
Oh,
Do you think that's
It looks, the rock looks good.
yeah, that's a tunicate.
Zoom
The
in
rock
there,
looks good.
video.
Yeah, if we're allowed to take the organism with it.
Yeah. Yeah, sometimes I'm not sure because they, you know, it looks like it's, you know, a good rock and it turns out it's just a big thick sheet of crust, but it looks like a good collection to me.
It has good angular vertices, I suppose, but
Yep.
until, yeah, I can't be 100% sure that it's a good rock, but I think it's good enough to collect.
Yeah. Well, we have opportunities to collect rocks for sure. We have lots of space. So I think we can stow it.
Roger. Nice tunicate.
Good. Yep.
Okay. Gibson on the
yeah,
the
B.
one that starboard box.
Yep.
So the one that says sample tray on your Herc hydraulics. So go to your Herc hydraulic page.
Okay. Sample tray out.
Yeah, and you're gonna want to usually hit 5 enter for me. And okay. Yep, bring the sample tray out.
So we are collecting a rock here in support of our geological science objectives. We last collected a rock about 700 meters below us, down deeper on the seamount, trying to collect rocks
Push
as we go up.
that box out a little bit further.
A little further out.
Yeah. What's in what? You can come up a little on that winch too.
Yeah, coming up.
What boxes do we have rocks in?
We have starboard bio box A and B available.
Roger. Think it'll fit, eh?
Nice shot.
Nice, yeah, nice.
Oh, lost the
Can
tunicate.
you do
Yeah,
Oh,
Cap
tried it.
Argus
Okay, you
there?
can close the box.
closing the box.
Great. So what did that go in, A or B?
Went in A.
A. Great.
Tragically lost the tunicate.
Come all the way in here, or am I going to hit the fingers?
Nope, come all the way in.
All right.
That was sample 9, right?
Correct.
That's why I turned that finger thing around so you can't hit him.
Yep.
I thought I hit auto altitude there, but I hit
You
auto
kept going
heading.
up. Yeah. How's that winch there?
That's good. You got 26 meters underneath you.
For those who are interested writing in, we're primarily going to be diving in the 1,500 meter to, 3,800 meter depth range for most of this expedition. There is a small possibility on the shallower end of that, or when we dive on Johnston Atoll, of seeing gold corals like Kula Mana Mana. But we will see as we get a bit shallower. I know it's pretty characteristic to this region, and it was originally described from the area around Hawaii here. But it's always very popular to see. They're very old, ancient creatures. One of my favorites. It's a type of zoanthid. But it's a
Sorry, Steve,
possibility.
you changed her back to the east again, did
We'll,
you,
we'll know when we dive on the atoll. It's probably the most likely place to see them.
Yeah, well, let's go 135 for a while longer. We want to come up the hill here a bit. Sorry, Lynette, I didn't catch that. We're just gonna go up the local hill here for a while. We're going uphill, so the uphill at the moment is 135. So we're gonna chase this up for a ways and see what we get. There was some debris here that is
Star, right?
typical of
There's a sea star on the center.
a lot of stuff above us falling
If we
down.
have time to take a peek at that.
Sure. Once we get to the top, we'll go back to the big bathy. You can zoom in there, Dan. It's a little tiny guy.
Yeah.
Oh, it's adorable.
So this is the other kind of sea star, not the Brusingid we were seeing down deeper, but this is a Goniastarid star, or sometimes they're called cookie stars.
Lost a limb.
There are many different, yeah, looks like it might have lost something or regrowth. But there's many different genera for this family, and usually when I guess, I'm always wrong, so I'm not going to guess, but I'll try and use the tools that I have to get a better idea on that.
Sometimes you gotta use the force. Okay.
All right, great, thanks.
There's a little collapse there. A little window. Oh yeah.
Another branch of something in the upper right too. If we could snap zoom on that. I don't have to linger, but quick zoom.
Yeah, go ahead,
It's not
Ben.
there.
Zoom in there.
Ctenophore, benthic ctenophore. Looks like another small bamboo coral. Yep, that's what it is.
Okay, that's good, thanks.
Nice botryoidal rock textures too.
Zoom back
Good
out.
crust.
Botryoidal? Botryoidal?
Depends
Yeah.
on depends on who. Yeah.
I always call it botryoidal, but
Yeah.
grapes.
Where'd you see the Ctenophore, Steve? I missed it.
It was just to the lower left of that. I think you might have to back up a little bit.
I think we might have blown him away there.
Yeah. It was anchored to the substrate. Yeah, so it was kind of down right here in that pocket. There it is. Yeah, he was blowing away there.
Always in the wind.
Mm-hmm. It looked like it was anchored. It wasn't moving too much, but it might have just knocked it off.
Is that it, that little orange thing?
Yeah, yeah. Looks like a Ctenophore.
Zoom in there.
Gelatinous zooplankton.
That's
See some
nice.
of the iridescence, the light refracting through the cilia that are on the inside of its body. It beats those to basically create a current to move.
It's flashing as LEDs.
Yep. The original LEDs.
The original LED.
They also have, in some cases, bioluminescent capacity, which we wouldn't necessarily be able to see right now. Our lights would kind of blow that out, but oftentimes in the shallow waters, even if you're swimming, sometimes if you see ctenophores and if you disturb them, they can bioluminesce at night, and you can see that. Great shot.
I'm hoping these photos are going through.
Yeah, they will come through. Sometimes they're delayed through C-Log.
Ah, gotcha.
But those iridescent rows are called comb rows. Those are basically rows of cilia that have multiple purposes in the animal. It was thought, or there's a point of debate about whether ctenophores or cnidarians are the more primitive organism, but won't go into that here. I don't want to get any angry fan mail. But they look very similar, but they're a completely different group than the cnidarians.
And I believe
Different
those
phylum.
ciliary rows are full of sticky cells, am I correct?
Right, yeah, in contrast to the cnidarians, they're sticky rather than the cnidarians, which are more stingy.
Nice.
If this one was anchored, there was a good chance it probably had tentacles that allowed it to anchor. which might tell us something about its identity as well, and which group of ctenophores it might belong to. There's two major groups, the tentaculata and the lobate ctenophores.
So do all ctenophores tend to anchor themselves on the substrate?
Not necessarily, some of them. And it might only be temporary. There's some ctenophores that have that exclusively anchor themselves to like a branch of a coral, and then will extend these like fishing long lines off of them and collect whatever happens to come through.
Nice.
Oh, that's pretty awesome.
We could probably put that in the highlight video.
It's a great.
Jonathan can do his magic and it's my favorite sea critter right there. Better come up a little bit there, Gibson.
It's coming up right on top of the mount there.
Yeah, that's where we want to be. It's a beautiful Argus view. Sorry, Atalanta view. Argus is on deck.
Looks like some more bamboo coral. Let me see. Chrysogorgia.
Might be officially the top there.
Yeah, nice little peak.
Dan, I'm
Not
heading
much
out.
there.
Roger, thanks, Dave.
Couple-hour nap. Dan's got it all. He's gone through all the different video scenarios, and he's a pro now.
Yeah, it's official now.
I would say
Absolutely. Take it easy on him.
Yeah, I'll try.
All right.
Let's see, which way do we want to go now? Let's go back to our, what were we, 040?
The original heading.
055, yeah.
I'm just hanging out in front of you now.
Yeah, I'm gonna come do a Herc selfie here.
Oh yeah, take an Atalanta shot with that.
Yep. Nice selfie.
Nice selfie.
Can tilt down a little as I come down.
Yep.
Okay, go back to the seabed. Enough selfiness.
We're still seeing, I thought that peak would be covered with more sponges and things, but we're still seeing a bunch of these sponge stalks.
Yeah.
You know, in some instances last year where we were seeing them where they were fairly heavily manganese coated, so very dark. But it doesn't seem to be the case here, at least what we've seen on our watch so far.
So you'll have to change your heading back to
Roger
to
that.
045 now.
Dead sponge stalks, do they have any scientific value?
045.
I assume they do.
Yeah, in some cases, so it's very difficult to date sponge debris material, you know, to determine the age of things, but
Can
I
tilt
mean,
up a bit for
the
me.
dead rubble,
Tilt it
not
up.
so much. I mean, there's probably more value of it to, you know, the biology that lives on the seafloor and the animals that live in and around the rubble. You find you know, they kind of multiply the biodiversity of an area. You might have worms and sponges or worms and crustaceans and other animals living in and around them. But there's not much we can do with the rubble itself. You know, to make a really good identification,
Coming up 5.
you would need to
That's all right.
also have all the parts of the sponge. You'd need, you know, the head of the sponge, because it contains different spicules than maybe the stalk.
Oh, is that a stalk crinoid that we saw earlier at towards the bottom of the screen?
Yeah, I think that was a black coral actually.
Oh,
Yeah. Didn't get a good look, but it looked like a black coral. But I'm sure we'll see more of them.
Come up a bit more.
Roger.
Bit of a wall here.
Yeah, that's a pillow there. Collapsed.
Wow. Yeah. Just completely filled in with sediment. Little sandbox, I suppose.
Total sandbox.
Anyone
Is that a
home?
sea star?
Hmm?
Is that a sea star? By that collapsed vessel?
Crinoid, yeah, crinoid. And it looks like there's a couple of xenophyophores in there as well. Single-stelled foraminiferan protists. Those are those nodules in the bottom of the sediment there.
Speaking of sand, do you know the actual grain size of the sediment? Is it sand sized or?
I don't know. I have a sample down in the lab that we can look at from other sites. It's pretty similar, grain size, from site to site, seamount to seamount. So we can take a look. It's fairly fine, but if you look at it under the microscope, you can see they're like these pearlescent orbs, very well formed.
Are they spherical?
Yeah, somewhat. Yeah, more spherical than irregular for sure. There is a fair bit of like silty mud too, but that'll wash out often. You'll see with the nodule sample, I think we scooped, that you'll probably have a bit of mud they'll have to wash out before you get that to the nodules, and then there'll be a bunch of granules of those foraminiferan shells too. But up on top of this local peak here, I've got one Chrysogorgia colony we just passed, as well as a few bamboos, similar species to what we've been seeing before. Nothing crazy, but it does look like the current might have slightly picked up as we came over the crest of this.
So it'll kind of a peak jutting out here.
Yeah.
Or,
You know what? Could we pop a Niskin right in this area just to be
sure.
safe, since we haven't seen much density here? This is probably the most we've seen. We've taken one Niskin so far, which is the one all the way in the back.
Right.
We can take another one here. We don't want to drift too far off the bottom, to kind of like right here or a little bit lower is ideal.
Right.
Or right next to the colony, if you can get it, even better.
I can.
Yeah, because we've got a couple of Chrysogorgia species here, a couple of bamboos. And I know for a fact we have some good vouchers of some of these Chrysogorgia already from other locations in the Pacific, as well as some of these bamboos, so it should be really useful to get an eDNA sample here.
Gonna blind you with the porch light there. So we've taken number one, you say.
Yeah.
Can I ask why they're called Niskins?
It's the person who invented them. Yeah.
Figures.
I think the Niskin bottle was invented in the 60s,
Yeah.
I think, which was itself a variant of something called a Nansen bottle. It was invented by a Norwegian oceanographer, explorer, back in the 1910s, I want to say. So just a different iteration of a different type of container.
Right.
There's that little swimmer there.
Yeah, it's like a polychaete worm.
Sure.
All right, so that's a good Niskin sample at 25, 43 meters in a stand of Chrysogorgia, a couple different species, bottlebrush and puffballs, as well as possibly declayed unbranched bamboo coral lips.
The DSC is turned on, Steve. So
Is it? They told me they turned it off because they were getting a ground fault, but I'll try and boot it back up.
yeah, should be running. I don't know what they're. Sorry, I meant to check earlier and I never did. It's the craft that has a ground fault, and I haven't bothered to sort of run it through its range of motion to get rid of it.
We're coming up on 0600. I'm going to get started on checks.
Roger.
I have a crinoid question for you, Steve.
Sure.
So those crinoids that we saw toward the beginning of our watch that were sort of hugging that stalk,
Yeah.
how do they find a stalk to hang on to?
Great question. I'm not entirely sure if there's something like chemosensory they use to detect, that substrate, or if they just kind of, meander their way in the dark until they find some substrate. I'm sure it's a slow process, and I'm sure once they find that that substrate that they like, they probably don't move. So it's a good question. There could be something that happens during settlement. Also, when they're larvae up in the water column, they're able to sense out some sort of, something about the coral or sponge there that they want to attach to, and then they land on it and just grow on it. So I think it's probably many of those different things.
Okay, cool. Yeah, I was just wondering because it's so vast, like without things sticking up into the water column, it just seems like to find it by chance would be impossible. But
yeah.
Cool, thanks.
Yeah, once they find a spot, they're not going to move, so they could be there for
5
years
meters.
or decades even.
Coming up 5.
But it is it's always a good question how they find their host. In the case of like associates, like if we look in some of these Chrysogorgia colonies, sometimes they're filled with shrimp or crabs that stay there for life. They'll actually settle in the colony and live their entire life cycle in the colony. There's probably some sort of signaling, either chemical or light sensing, or bioluminescence sensing that larvae might use to find their host coral. But you'll find sometimes too that the eggs cases are laid right in the colony by crabs or shrimp, and so the young might not even go very far at all. You might have several generations in one coral or on one branch.
Awesome. Thank you. Yeah, that makes sense. You find something good, hang on to it, right?
Right. I'm trying to connect to the DSC, and it's saying it's not available.
Let me I'll power cycle it, and it'll take probably 30 seconds at least to for the NUC to reboot.
Steve, do you mind if I read the actual chat?
Yeah, sure. Throw us some questions.
Craft comp, Dan?
What's that?
Which one is that?
It's the
Far left?
yeah. It's,
Oh,
see the big orange band there?
yeah.
It's hidden by the number 4. It's not a gauge, it's a physical comp.
Oh, I understand. Thank you. Okay, Herc comps look good.
Dropping off the ridge here down into the ridge is off to our left a bit there. I don't know if we want to stay up on the top of the ridge or yeah, I think so. It's flat for days. Yeah, let's go 50 meters north now. It's all flat and rubbly and boring down there.
They'll grow up big and strong.
Oh, there's something moving on the right side. Brittle star, maybe.
Yeah.
If it is what I think it is, it looks like a brittle star and not a Brusingid.
Does look like a brittle star.
Even though it has similar color, the Brusingids often don't move like that, but the brittle stars will. And they'll often do this.
They'll
Fall
jump
down.
off.
Tumbling.
Oh, there's a spinning snail too.
Zoom in there a bit if you want video. Spastic snail.
Yeah. So this is a type of snail that uses a very unique way of moving across the seafloor.
Might be an understatement.
That's good on the zoom there.
Man, he's moving. Look at that.
Zoom out just a bit from, yeah.
It's a gorgeous
You
shell.
just keep the lasers in there. I can.
The genus of that snail is called Gaza, G-A-Z-A. And we see it from time to time, oftentimes headed in one direction. This one's kind of stuck. Usually they just go down slope.
Oh, here we go. Here we have a question. What is your favorite Holy Grail slash favorite slash Holy Grail coral mystery that you hope gets solved?
You know, people ask me this all the time. Questions, you know, like
Okay,
that, like, you know, why do corals get their color? And I feel like I don't have a really good answer for that. So, you know, where do deep water corals get their color from would be a pretty great answer to solve, or question to solve. In part because it might tell us something about, you know, how they how to delineate species, for example, if color is something that's maybe not as flexible as we previously understood. That would be a pretty big one. Yep.
Okay, checks are done. Comms look good. Temps look good.
Roger.
In contrast to a lot of shallow water species, which get their color and pigments from zooxanthellae, the symbionts that live within the tissues, a lot of deep water corals don't have those symbioses because there's no light down here for those symbionts to survive. And so what you end up having are just kind of really strange color patterns that emerge. Reds and yellows and blues and purples, and it's not quite clear what they're using those pigments for. Another sponge stalk here. Decapitated.
Totally decapitated.
A little morbid.
Couple of
Zoom in there for a second if you want. Kind of waiting on.
Oh,
on
the
Monte.
humanity.
What is that white thing attached to it?
looks like another brittle star, possibly an Ophiocanthid.
Is it just eating it, or?
No, oftentimes these brittle stars will just stay there for, they'll use the dead sponge stalk as substrate. So these are probably filter suspension feeders, so they'll find a spot and they'll stick their arms up into the water column and use those spines to presumably grab particles that might drift by out of the water column and ingest them.
Yeah,
And we see these often on corals as well.
I'm too far away.
But that's typically what they're doing. Then you have others, like the one on the rock to the lower left, that are probably deposit feeding or in transit. And deposit feeding brittle stars will often ingest the sediments or something in the sediments and extract nutrition that way.
We have another question coming in. Says, what's the largest living thing you found on one of your missions?
Personally, I think it's probably the sperm whale we saw in the Gulf of Mexico in 2015.
Oh, wow.
That was a pretty big moment. There's something bright white down there.
And I gotta say, since this is my first mission, probably a sea cucumber that was found maybe 15 minutes ago.
Same here.
What do we got there? Is that a fish?
Hey!
What's that?
Oh, it's debris?
Yeah.
No!
Plastic?
Trash, yeah.
Oh, it's a flip-flop.
Oh, yeah.
It's a sandal. Yeah.
That's depressing.
Somebody lost their sandal. Only one, too.
Somewhere around there's a one flip-flop to...
What size is it?
Well, we can actually
you can
determine
grab
that.
it and get you a new pair.
That'd be great.
Decayed flip-flop, my favorite.
You know what? I'm surprised that. Well, I know you might have to correct. So it's foam, right? So it's going to shrink. So you're going to have to correct for the shrinkage that occurs.
Oh yeah.
So.
Surprised that it sank.
It's drifted a pretty long way.
Yeah.
So you're saying if I'm a size 7, that's a size 10, I could probably fit it.
I don't know, you'd have to do the foam shrinkage calculation.
The foams. I'm sure there's an equation for it. Foam shrinkage per capita.
Yep. Even being, what are we? 600 miles, 800 miles, something like that, from the nearest major population source. We're still seeing a lot of marine debris, although I think that might be the first for the dive.
You moved that way. Atlanta has done.
But it's not unusual to see marine debris.
Nothing, so.
Some of the more common
wanna
things
come
are
up
soda
just a bit
cans
there?
or paint cans
Come
or
up 5. Give
stuff that might have been thrown
up
off a
5.
ship at one point during transit.
Oh, goodness.
All right.
Atlantis. I don't know. It's really unpredictable, that whole thing. Didn't realize how spoiled I was with Argus. Just when I think I have it figured out,
Is that another sponge stock?
Yeah, sponge stock there. Can we look a little bit behind the sponge stock at that
sure.
puffball?
Cool.
Zoom in there a bit, yeah.
I'm sure there is an equation, we just don't know it. Is that bubble gum?
Chrysogorgia?
Yeah, this is a golden coral. It's a Chrysogorgia, yeah. This one is in the genus Chrysogorgia. It has one chirostylid squat lobster associate. Oftentimes you find these associations. Very rarely would you find the coral without its most common associate.
Okay.
And believe it or not, the associate can actually be used to identify the coral because they have such high specificity at some places and some depths that if you can collect the associate, you could almost always identify the species of the coral.
Wow.
But as you get shallower, we find that the associates are more flexible with the colony they can inhabit. You find many different species on one type of coral.
Get a full zoom on
Something after we get the crab just to the lower left of the frame, there's another small bit of something, looks like an invertebrate, possibly a predator.
lower left.
Yeah, or lower right, sorry. Yeah, right here, is that might be an amphipod.
Okay, I can push it on that. Let's see what he's looking at now.
Or, actually, it's yep, it's an amphipod. So amphipods are crustaceans, close relatives of those that isopod we saw earlier. But oftentimes we'll find these in and around the colonies as well. They're very hard to ID though without collections. All right, great. Thanks for the zoom on that. There's something up and to the just directly upslope from where we are that I was kind of curious. I wanted to look at.
On the left of the coral too, there was a smooth rock face. Looked like it didn't have any crust on it or anything.
So what would create that?
I assume just erosion happened rather recently.
What did you want to see, Steve? The stock there?
I thought it was this thing, but I'm going to pass. It's just a sponge stock. I just didn't see the entirety of the thing. Wasn't well lit at the time. So we can pass on that.
Got it.
Ooh, another glass sponge? Or is that just a stock?
I think it might have been another stock, yeah.
The one to our left there?
I'm not seeing a huge number of live ones here.
What does that mean? If you don't see a whole lot of live sponge, does that mean this is just like there's a lot of predators around, this is not a very nutrient-rich area?
It's, there's another colony we haven't seen yet here, down on the bottom.
Oh, yeah.
So it could mean a few things. These sponge, debris and sponge rubble is persistent through time. So we could be looking at, a die-off of that colony sometimes, years or decades ago. And it's just, it's been an accumulation over time, or longer, It doesn't mean that, everything died here and that doesn't exist anymore. It just means that there's a lot of persistent rubble. This is everyone's favorite tongue twister coral.
I'll zoom in there, Dan.
What's it called?
This is another similar species in the same family as the Chrysogorgia we saw earlier.
Chrysogorgia.
This one is called Remulogorgia, Remulogorgia militaris. And this one is actually colonized by a fair number of other non-coral things like anemones here. These are the actual polyps, these fine white structures, but there's some anemones here. These are parasites or something that takes advantage of the colony. You know, whenever there's bare skeleton like this, you'll get things that attach. Usually indicates, you know, that there's some of these worm tubes as well. Usually indicates that, you know, the colony is probably on its way out. You know, it could take years or decades or longer, but
Got a wall pretty close. I'm gonna
oftentimes when they get completely colonized, it means they're
Okay, I gotta make
not able to fight back against potential predators. So if you're a coral, you want to make sure that you cover all your you know, bare skeletons so that you get nothing that settles on there and starts competing for space. Because the animals that are competing against you are feeding off the same things as you are, like you know, the particulate matter, marine snow, small invertebrates in the water column.
They're probably better at eating than you are, so.
Yeah, they're bigger, you know, larger tentacles, probably have bigger defenses. There's a live sponge right there. You can just see it looks more vibrant, translucent color.
Can we get her to hold position for a few minutes? I'm not sure. Feels like it, but I'm just gonna come back out in front here. I wasn't paying attention to Gibson's looking west now.
Want me to swing back around north? Or?
Yeah, I'll come around.
Okay.
Usually I catch it when you change your heading around, but I did not that time.
Copy that. Thanks for that. Just coming up slow.
All right, I've got the DSC up and running again.
Nice.
Oh, we got another type of glass sponge there.
Do you believe in the megalodon or any other sea creatures? No. No, I don't.
Believe in them?
Yeah.
Well, I mean, it's a fossil.
Well, I mean that they exist currently. Like the Loch Ness Monster.
So this is a euplectellid sponge in that family.
particles in the water, are they organic or minerals? I believe that's detritus. Am I wrong on that?
Yeah, it's
zoom in there a
it
bit,
is, you know, it is mostly organic.
That's good. Thanks.
Still, we can say that, but it certainly, you know, has less organic material than when it was up at the surface, or, you know, shallower.
Yeah.
So this is a euplectellid sponge, probably in the genus Regadrella. It has a couple of associates that live inside. These are usually shrimp that live inside, and they're stuck there. The sponge, they'll often, you know, live in as larvae settle inside the sponge. The sponge will cover over those shrimps, or sometimes crabs in there with the sieve plate, but they're effectively trapped, and they can live off of exactly what they have inside the sponge, usually detritus material or things like that.
Are the shrimp parasitic, or is it like a symbiotic relationship?
Yeah, it's probably a commensal or some sort of symbiotic relationship. Yep.
Can't quite get the shot I want. Come on, Herc. That's the money shot there.
How's that looking?
There you go. Screen grab.
Did you get it?
It's got it.
Yeah,
Good.
got it.
Get it, got it, good. We're out.
All right. I've got the DSC tuned in, plumbed in, so it looks good from here.
So
If you see anything worth flying around, try and get some photos with that. But I don't think we've seen anything too big yet.
should be, Dan, you should have, as a PC source, you should have, what's that PC called?
Are the sponges made up of single-celled or multi-celled organisms, or multi-celled organisms living as a colony?
Actually, see if I got it here. Monitor.
So they are multicellular, although they're made up of a lot of different cells that have very specific functions. Like some cells might be, you know, designated to transport nutrient throughout the sponge.
Try it again.
Other cells might be directly related to only creating a feeding current. So there's a lot of specialty jobs for one multicellular organism.
There is a way to put that up in one of those unused boxes up there.
Okay.
We can't do it. Dan has to do it.
So it's a still cam on my left monitor. I want to try and get it up in one of those.
Gotcha. Do you know what that still cam name is? Or?
It's a PC source called StillCam.
Okay. Let me see what I can do.
It's under one of your, bring your head to the right, will you?
Coming over.
Give me a second. Can bring Atlanta up a bit. I'll use its
Up.
sonar there and see where.
Looks like we have all three flow types in one picture right now. You can see some pillows, the smaller ones, some lobate, the bigger ones, and then some sheet flow as well.
How do you think that came to form?
Yeah, Gibson, if you don't mind, I might wait for Dave to come up. Just different
Show me how to get that pip. I just don't want to mess it up
rates
for you.
of the magma. The slower the rate, the more bulbous the feature.
Let's try 50 meters back on our original course up the hill there. 040.
But now we're just in all pillows, it looks like, all pillow basalt right now.
Which one is it? Oh, okay, almost guessed right.
Oh, it's another Ctenophore.
Mouse button.
There's its tentacles.
Same Ctenophore, yep. And you can see the tentacles now a little bit more easily. And they're completely retractable too. They can actually draw their tentacles back in.
Zoom in just a little bit for me, Dan.
Like a tongue, I suppose.
Mm-hmm. Yeah, yeah. And it will, you know, if there's any food particles stuck on there, it can ingest that.
Okay.
We have another question from the chat of how long has Mr. Cormany been piloting Hercules?
Since 4 o'clock this morning. My first go with Herc was in 2018. I don't know if I can reach Steve.
What is that?
There's a few things here.
I'm gonna
Spin me around here.
oh, the ship's moving that way. We'll wait for it.
All right.
We've got a couple things. If you want to look at something close, this thing here is of interest.
Roger. Okay, Dan, you should be able to push in there on it. Get the dramatic black in the background.
Beautiful.
Yeah. So this is a primnoid. I think it's a new species for this dive. This looks like it's probably Narella in the genus Narella. Probably Narella macrocalyx, based on how it's branching and the sparseness of the branching, the size of the polyps. This particular species has very large polyps compared to many other primnoids, but usually has this kind of very sparse, very low branching morphology. So it could be one other species that was recently described as well, but collections would be needed. But I think for the most part, probably pretty safe in going with Narella and possibly macrocalyx
push
for
right in
this.
now, Dan.
Yeah, there, that's great. You can see the individual sclerites even in the body wall of the polyp.
Come on, Herc, hold still.
Another question from the chat. How much of the ocean floor would you say roughly has been explored?
Okay, we can go
Or
with
roughly
it.
that we've explored?
Yeah, there's always the counter question of like, what tool do you want to evaluate? I mean, do you want to use like mapping? It's probably somewhere in the 10 to 20% mapped in terms of eyes on the seafloor. Maybe probably a percent or less. Even though we go out here for 2 or 3 weeks at a time, we're still making very incremental gains in getting eyes on the seafloor. So it's probably a couple of percent or less that's been explored by human eyes.
And the Nautilus has explored that, like less than 1%?
Yeah, a fraction of a percent. It requires a lot of effort from a lot of different countries and a lot of resources to do this type of work. But it's very valuable.
So what is this tissue paper looking thing?
Bring
we're
that
looking at
DSC
right now?
back up. for
Yeah,
me.
so what we're looking at here is a
The
different
digital
kind
still
of
can
glass
look up.
sponge. Different species of
What's
glass
the
sponge.
name here?
Looks like it kind of blew
Still
up
can.
there.
Go for it.
This is actually a healthy looking one. It doesn't look too degraded. I think this is a type of Pherididae in the genus Pherididae family. Genus, I'm going to double check on. Within the Pherididae family, there's a tremendous diversity of genera and species.
It's
Worth a second look.
It's framed up in the DSC there, Steve, at the moment.
Yep, I'm swapping over.
All right.
While we wait, I have another question from the chat of how many new species have you discovered, and did you get to name them?
Myself, none yet. Currently I have one in progress.
Oh?
But it hasn't yet been named, so I don't want to give that away.
Can you give us like spitball ideas, stuff that have been thrown away?
No, not quite. It wouldn't make any sense.
Keep your secrets.
Yeah, you can read about it in the papers.
When I'm rich and famous.
All right, I got a few. I'm having this
I
weird
see
halo effect. I don't know if it's. I've got like the
Yeah, you'll have to crop that
you see
out
that?
of there.
Yeah,
I can try and
like a
hold
like
it
a
here
light
if you want
halo.
to play with it, or
It's not the housing. It's like a light halo.
see. Try it now. I changed the lighting.
Still
doing
there.
it.
I mean, it's a good photo. I can crop it out.
Gotcha.
All right. I got a few good photos there on DSC. So this is a Pherididae.
Sure.
And is that a fish floating to the
Yeah,
left of it,
you got a
or is
little
it like an
Something
eel?
behind it.
Oh yeah, there's a fish. Yep.
Yay! Just for you, Asako. There's your fish.
So this is I believe it's a type of cusk eel.
A cusk
Need
eel?
to come up to get over
eel,
that ridge.
yeah.
You're all right.
Couldn't do much better
It's a good shot.
right now, unfortunately. A lot of the characteristics we're looking at for fish identification are like morphology of the lateral line and morphology of the fins. And sometimes that requires some fancy flying because oftentimes they will always want to orient away from you. Maybe it's the lights. They don't like a big noisy robot in
They're
their
camera
face.
shy, we understand.
There's
Yeah.
a reverse angle on the DSC too.
Alright.
Have you ever been tempted to see what the specimens taste like? Absolutely. Some of these sea cucumbers look delicious.
Yeah.
I'm gonna leave that to you, the tasting.
Tempted, yes.
You know, when I start convulging on the floor,
Committed,
we'll know.
no.
I think one was called the grape jelly.
You know, I'd love to see if it lives up to its name. I just don't think I'll be around to see the result of it.
I'd rather lick the basalt than taste the sea cucumber.
Why would you do either?
He's a geologist.
Has your team explored the Marianas Trench? And if not, do you want to?
Not yet, and probably, yeah, maybe someday in the future, near future.
I've seen some stuff out of the Mariana Trench, some of the teams that they've sent down there, and there's some ugly fish down there.
So are you changing the lighting, or is that just me adapting?
What's that?
Have you been changing the lighting? Or?
That was me, Gibson.
Ah, okay.
Yeah, I got your iris over here.
Cool.
I'm just giving you a better picture.
Yeah, very cool. So that's all your controls there.
Yeah,
Raj.
this is your iris zoom.
Oh.
Focus right here.
Speaking of that. Yeah, okay.
How's that for the wind right now? You want to up?
Yeah, it's a good shot at the moment. Yeah,
Oh, there we go.
come up a bit if you want.
Yeah,
I'm coming up here on the hill.
Raj.
Is
Oh.
that another cuskeel?
Nice eye.
Yeah, I think also.
He chilling.
Do you think it may have been the same one or a different?
It could have been the same one, yeah. Oh, you know what? That actually might have been a different family of sponge. That could have been a Tretopleura. They all look very similar. So I don't bother guessing with sponges anymore. It's usually learn that I'm wrong later on.
And is this another octocrawl that we're
Yeah, so we got here, this one looks like it could be another Narella species, but we'll take a
Can
quick
push in a
closer
bit there
look.
if you want to.
Copy that.
So it's a primnoid octocoral. Likely in the genus Narella,
That's good. You can hold that. I'll
based
get a
on
little
kind of
closer.
the bushiness and density of the branches. So it's got a number of anemones on it as well. These anemones are probably parasites taking over any bare patches of skeleton they might be able to come across.
So can they grow out and start just entirely engulfing the coral, or?
In some cases, yeah. So if you see like in the one on the lower left here, or in the far left-hand side, they'll often wrap around the colony. However, there's some instances too, like here and here, I believe, where the coral can fight back in some cases. So what they'll do is, like right here, I think there's one, and right here, if there's an anemone that attaches, the coral may be able to grow up and around, and then basically encase it into a type of gall, and then the polyps will grow over it, and oftentimes you'll see structures like this, like lumps in the colony, and since it's covered with polyps, we can suggest that the coral probably won that battle against the invader.
Nice.
Do you reckon any of you could swim down there? No, I like my insides being inside, not outside. Have you ever accidentally killed something with the Hercules? Have we? Sorry.
Okay, you can come up a bit now. I'm underneath you.
It's like a little
I'm
arch form.
I'm behind you.
Right there.
Coming up a little bit.
I need to move ahead here.
Igneous arch.
Roger.
Rare.
Delta's 15, are you happy with that?
Yeah, I got 50 meters under you. I'm going to try and come back to the northeast a little here.
Okay.
Some more sponge stalks.
More sponges, yeah. So this one here is also another type of sponge. This one looks like...
let's hold off for a second.
This one looks like it could be in the family Pherididae, with like a PH, P-H-E-R-O, and
Is that the sea cucumber that was down to our left that we just passed?
yes, yeah, similar to the one we had collected.
Yeah.
And the genus on this sponge is called Poliopogon, P-O-L-I-O-P-O-G-O-N.
Here's another question. What is the white stuff covering all of the rocks?
Yeah, good question. Good question. That is probably sediment. It settles into little
There's a
pockets
sea cucumber, eh?
into the substrate
Right off your
between the
front
crusts. And oftentimes you'll find that sediment will accumulate in areas that might have stagnant or slower flows than in areas that have very rapid currents, which tend to keep the sediments off the substrate.
Once the sediment gets there, though, it stays for a very long time, for the most part, until the rocks get subducted, usually. Or we disturb it, I suppose.
Tilt up now for me.
Coming up.
So definitely starting to see more diversity at this depth, where we're at 2,400, which is kind of what I expected for this area, and it should increase through about 2,000 meters. There is like a deep bathyal diversity maximum, I've come to call it over the years, although it's not an official term yet, but that just means that in the bathyal zone, which is between 200 and about 3,000 meters,
See that loop in our tether there?
you have
Yeah, I see it.
peaks and troughs in diversity as you go down and with increasing depth. And right around in this region of the North Pacific, between 2,000 and 2,500 meters, you usually run into a peak of diversity, which may decline slightly as you go shallower, but then typically you find another peak in diversity somewhere shallower, maybe around 1,000 meters or 1,200 meters, something like that. So we can explore those hypotheses as we continue to conduct this dive. But this is pretty characteristic species and diversity patterns for what we expect to see. Although I think we can probably still see some more abundance as we move shallower. Things are still pretty low density, corals, sponges. But with the right conditions, for example, the right flow conditions, which seems to be picking up here with kind of a north to south
No, I just pulled it out.
current, you can see based on how the marine snow
Yeah, because
particles
that one wasn't recorded in the
moving
racks,
across the screen.
So just a little.
Let's try a 20-meter move east. See what happens.
So question for the chat, have you found any octopus yet? I don't believe so, no.
Not yet.
Not yet, but they're there. Probably, we'll see at least one or two on this expedition.
Now, would they be considered more octopus would be more common or squid?
Octopods would be more common at this depth than squid. Yeah, at least on the benthos. Yeah, there may be other squid in the deep water environment we're not seeing, but on the benthos itself, octopus, like Dumbo octopods, are more common.
Another question of, what is the weirdest thing that you found, like a blobfish or etc.?
Sandal.
Yeah,
Sandal's
sandal.
pretty weird. Trying to think of all the trash and debris I've seen.
I remember that cracked helioclastite, the yellow brick road. That was pretty weird
Yeah, that was.
in terms of geology.
Yeah, there's another glass sponge there in the background.
Just,
So
we're
okay.
gonna position
There's
Atalanta right over the ridge line here, Steve, and then we'll start moving up the ridge again.
Yep.
Finally figured out which way I want to go.
There were some weird things
For the
we
next
saw
few
last year, end of last season. These kind of white egg-shaped rocks in the middle of, 3,000, 3,500 meters, something super deep. And they were totally enigmatic because we collected one, brought it on board. It was carbonate or something like it, but you don't find that at 3,500 meters, so it's probably dropped there, we concluded. And it turns out it was probably some debris from like an old guano mining expedition, some ballast or something that had been dumped, because there was a high amount of phosphorite, I think, or phosphorus in the sample, in the rock. Suggests it's not carbonate, something else. So maybe it was mined guano that had been dropped from ships that had passed through here 100 years ago.
And it never breaks down.
Yeah, it was interesting. It had a very like milky, powdery texture. So but it wasn't dissolving by any means. Yeah.
Sea cucumber around too.
Still
About
fairly
4.
solid. Sea cucumber.
Yeah, I'll let it swing in for a minute here, but yeah, I think 045. So I've kind of come on both sides of the little ridge here, so I'm going to try and chase the ridge up, and we can hop on either side if we see anything. I'll come back over on the left side though. Bring your head back around to east, northeast, somewhere in there. Okay. Yeah, ready.
So Atlanta was being used instead of Argus. How is Argus doing? Argus is currently just stored on deck, I believe. Just kind of hanging out, waiting for the next mission.
You could make the argument that Atalanta's actually just hanging out and Argus is doing nothing.
Is that an organism right there on the rock?
Yeah,
Little white.
it looks like, so there's a crinoid there, but there's also, yes, and can we zoom in here quickly? Quick snap in.
Yeah, go ahead, Dan. That's good, thanks.
There's a couple things here. This looks like a barnacle, stalked barnacle. And this is a small primnoid, possibly that same Narella we saw earlier, but a very small colony. If I was gonna collect that, I would want a little bit of a bigger piece. That one's pretty small, so I'll leave that one be. But
okay,
barnacle, yep. All right, there's another coral species we might not have seen yet, coming up right in front of the lasers. Glad we got that rock when we did, because it doesn't look like there's very much in the way of
everything's consolidated.
Loose stuff here.
push in there a bit if you want.
Is this another golden coral, maybe?
Yeah, I thought it was something else, but it's also another golden coral, Chrysogorgia. I think it's the same one that we saw before with these squat lobster associates. Yep. It's doing a very fine job of mimicking another type of golden coral called Metallogorgia, but based on the branching sequence that's coming off of the lower part. Oh, there's an egg case right here. Unclear what that might belong to. It looks like there might be one egg case here, right in front of that squat lobster, and then one here. I'm not sure if those belong to the squat lobster or if they belong to maybe some other animal that lays eggs in these. Sometimes certain snails will lay egg cases in these types of corals as well.
So the squat lobsters got their name because they're squatting on the actual coral, or?
No, I think it's just that they, so that if you were to stretch them out, they look kind of normally like a crab or a lobster, but they tuck their tail under their body, so it looks like they're squat.
Nice.
Compared to a regular crab or lobster, but they're very closely related to hermit crabs. Thanks, front row. Same group as hermit crabs and spider crabs. Not the true crabs that you find, in the coastal ocean. Although there are some brachioran crabs you find around here too. Although probably not this deep, maybe a bit shallower. Looks like the current's picked up a little bit around here.
Yeah.
Out of the north. That's exactly what the model predicted.
Okay, I need to turn around and go the other way. It's not working here. I don't know what happened to our ridge we were following, but it is gone. I give up.
It looks like it might have trailed off to the south.
It did. I was chasing it to the south, and it's like going the opposite way we want to go.
We can go towards the south or southeast if you want to reacquire it.
Yeah, can do.
We're making good time.
Yeah, you want to... the direct uphill at the moment is due south. Yeah. What can I say? No, let's do 50 meters south. Those, what is the resolution of the bathymetry at this depth? 100-meter grids, or?
Where are people tuning in from today? If you click on one of those tabs, the next one over, that one.
Trends. Great.
Yeah.
Most people are from YouTube. But in terms of countries?
Yeah, there should be a tab that says...
Majority from the US, about 106 from the UK, 93 from Canada. They're watching you from home, Gibson. 50 from China. And then see from France, India, Italy, Russia, Netherlands, South Africa, all over the world, really. From the Isle of Man, too.
We'll be here all week. Well, the next 3 weeks, actually.
So,
Yeah,
remember our voices.
That's a local high ridge there.
Yeah. Can we drop a target here and just call it a peak of...
Peak of confusion.
Local peak.
Peak of confusion.
We're trying to figure out, like, what is going on? I keep thinking I'm going the wrong way or something.
The heights of confusion.
Yeah.
Now, is that a piece of coral that we're seeing, or is that a sponge?
Oh, 700 down, I'm going to start gauge
That's
checks.
a sponge. That might be similar to the one we saw before, the Tritopleura sponge, but it's tough to tell with these large, long blade, fan-shaped sponge species.
Can't get there from here. I might be able to. You come down 5 meters.
Coming down 5.
Pull you around a little bit just to go have a peek at this guy.
Let me know if you want more.
And come down another 5.
Coming down.
Turn your auto heading off for a second.
Auto heading off.
Yeah, this one actually looks more like a Pherididae. Sometimes it's really tough to tell. But it's definitely a glass sponge, like the others we've seen. We don't typically see demo sponges until we're much shallower. Occasionally, but usually they're much shallower. This looks like it could be a Pherididae, but the morphology is kind of odd.
Okay.
I got some still cam shots there.
Oh, cool. I wouldn't even watch it.
Oops.
Get our tether back off the rocks. Okay, you can come back up a bit now.
Coming up. Can I go auto?
Sure. All right, we're gonna have to go downhill somewhere. Might as well go the way we're supposed to be going.
Yeah, sounds good. So we're
Let's kick her back into
waypoint
what our overall, whatever our bearing is on the way up to the top. Yeah. Shoot. Yeah.
We're expecting it to get more lumpy, kind of like this as we go up slope too, from waypoint 4 onwards. Like local hills and valleys.
So are we at waypoint 4 now, or?
What's up?
Are we at waypoint 4?
Almost. A few hundred meters, yeah.
Okay.
Maybe a little bit more than a few hundred meters off of 4. We had some expected kind of in the saddles of the ridge, you know, where you have a peak and it drops off. You know, we kind of expected to see, you know, more accumulation of sediment and then maybe nodules. But it seems like we saw that a bit deeper, but we've lost kind of the nodule fields, and it's good that we got some of those nodules. I mean, these might be bits of nodules too, but it's tough to tell if this is kind of consolidated here, or if it's just.
I think it's consolidated. Maybe.
Can we poke the sediment here?
Yeah.
The nodules. See how deep it is.
Come down 10 meters.
Coming down 10.
But yeah, it is good to note that, you know, you still have pockets of these, because I hadn't seen these until just until we said we hadn't seen them just a second ago. I'm gonna guess that these are loose.
It's probably a good guess.
Although you never know.
One way to find out.
Can you try and poke right in front of the DSC so I can try and get that on DSC shot?
Sure. Can get the manip to cooperate. Yeah, they're loose on the scoop.
We already got a nodule scoop a little bit deeper. I think it's good to know that they're here, but I don't think we're going to take a nodule sample here. We have one from a bit deeper, and we'll save our last scoop for a bit shallower, since we have some more of these saddles up shallower too. And if there are any nodules there, we can get a sample of nodules from the bottom and from the top.
Roger. Let's put that on the personal sample for later. Uh-oh. Really?
Well, I guess it's a little more personal now.
Wow. Alrighty then. See if Jess finds this later. It's a present for you, Jess, if you're watching. It may or may not survive to the surface. So I think I'm really there. I think. Yeah, everyone's looking east at the moment. And we're kind of in a little bowl here.
So can we drop a target here as well and call this like patch of nodules or something to that effect?
Nodule field. It's a pretty clean break between the nodules and...
Yeah. And they all start out as a core of something. I'm interested to see what, usually when we have the nodules last year that we saw that were this size, the core was like sediment or, grains of sediment.
Right. Yeah, the crust has to accumulate on something. Doesn't have to be a rock though.
Right.
Speaking of rocks, looks like something that fell
Max
from
tension is
above.
7,200.
This looks like a beautiful bamboo coral.
You have to minimize that. You have to use this and window that. You don't have a keyboard there. I don't know where it is. It's in here somewhere. So the Python script here.
And is that a glass
60,
sponge behind
80,
that
whatever, yeah.
bamboo coral?
Yeah, that one is glass sponge as well. It doesn't look very healthy. Usually when they're healthy, they're vibrant, translucent, or white in color. And then they turn opaque or dark as they start to die, because they get clogged with all sorts of debris and bacteria and stuff taking advantage of that space, breaking it down, discoloring it. But we're still seeing similar species, these large unbranched bamboos.
What's that?
Chrysogorgia.
Yeah, I need that camera at the moment.
A couple of different
The
species.
reservoir's good.
Oh, these nodule
I
patches
use
are
it when I'm
a few meters across, maybe.
to tell where the
Out there.
hell of the vehicle is there.
But really
Which
just
I'm not
breaks
coming
in these
You
sheet
can
flows.
click on this too.
think it's interesting too that
Yeah,
the
for
nodule
sure.
patches occur in the depressions where you would expect sediment to accumulate and,
Yeah.
kind of smother them.
Not usually the thought process that I would have to have nodules there, or nodules.
Okay, gate checks complete.
There has to be
Looks
good flow in whatever
Right.
area that is, so the nodules don't get smothered. So there must be some sort of currents that maybe excavate or keep sediment from building up in there.
Come down 5 meters if you want.
Coming down 5.
Yeah, we're not really concerned about the max tension now. We're well within the limits. So
Roger. What is the limit on the 6.8?
we have Atlanta on there, which is significantly lighter. B, we are above 3,000 meters, and C, the weather is pretty good. So
Copy that. Do we know 6.8 max tension?
as a general rule, anything in the teens, it would be concerned, and it would depend on the weather too, how hard it's heaving. So what kind of, it takes snap loads. Like that one we saw on the launch, were you sitting up there on the deck?
Mm-hmm.
That one. We can probably do some longer moves now, Lynette, if you want. Looks like it's going to be
There's another black coral
sonar saying the same.
off to right here.
Oh, wow.
Very dark silhouette. It's good to note that, that even though nodules, there's not a whole lot of things that attach to the nodules because they're relatively unstable. When you have a coral like this, it can be, easily knock itself over. But on the substrates around or near, sometimes within the nodule fields, you can get corals like these that grow. So this looks like heteropathes, what we've been referring to as heteropathes americana for a number of years. But this is the second colony we've observed on the dive so far.
So what makes a colony? Is it just more than one, or is it just, what are the characteristics of a coral colony?
The polyp is the basic unit, so multiple polyps.